Perfect.
All right, can everyone see the presentation okay? Awesome, okay. I will move the toolbar here. All right, let's get started. I'm Heather Chamberlain-Irwin. Thank you for joining us this morning. I am co-advised in the Matt Hufford and Andrew Somerville labs. Many of you know Matt Hufford, maize geneticist. Andrew Somerville is a biological archaeologist and anthropologist, and we're stationed here at Iowa State University. So what I'm bringing you today is my grad talk, and this project is in the really early stages. I'm also really early in my learning. So thank you for allowing me to share this with you, and I look forward to your ideas and feedback as I get this project off the ground. The project is entitled Genetic Legacies of the First Ancient South American State and Archaeological Maize. I should point out what a state is. According to David Sandiford, a state is a socially stratified and bureaucratically governed society with at least 4 levels of settlement hierarchy. So think capitals, cities, villages, hamlets. So really, our study is about social and political change. All right, so the plan today, I'll tell you a little bit about myself. We'll go through the introduction, background, region, question and hypotheses, materials, methods, testing those hypotheses, and broader impacts. A little bit about me, I'm a first-generation student. This is my 2nd year in the PhD program, and I'm in the Ecology, Evolution, and Organismal Biology department. I have a Master's of Science Education from the University of Northern Iowa, or UNI, in 2015, and a Bachelor's in Biology with a minor in Religious Studies, also from UNI. My work experience, I spent about 5 years working in an environmental lab, and this was part wet chem, part mass spec, ion chromatography. I learned a lot of different methods, and the last year as a quality assurance assistant. And then I spent about 5 years as a high school science and computer science educator, and I left because I wanted to pursue a PhD. And a non-sciencey human-y thing about me is I started a sustainable soap business. After my PhD, I plan to work for a nonprofit as a researcher or curator for germplasm diversity with a special interest in Indigenous food sovereignty. I'm just gonna pause a minute. I think not everyone is muted. I'm getting a little feedback here. Could you take a minute, make sure you're muted? Awesome, okay, quiet on my end. Thank you so much. All right, so let's introduce the project. What can ancient crop DNA tell us about early human communities? This is really an interdisciplinary plant genomic and archaeology study. Specifically, I'm looking at evolution and diversity of maize in Peru. The region in Peru is Moquegua Valley in South Peru, which has 3,000 years of documented maize cultivation. And another interesting thing about this region, it was settled by the first people from the first state in South America, the Tiwanaku. We know maize is socially and politically important and it follows people. So what was being selected for? And can we see those legacies in Peruvian maize today? Well, we can't talk about maize without talking about maize domestication and dispersal. So maize was domesticated in the Balsas River Valley in Mexico about 9,000 years before present, or BP. It dispersed quickly to South America, which is considered the secondary improvement center. Upon arriving to Peru, maize encountered a genetic bottleneck and extreme environment of the Andes. What's interesting about this map is it displays only directly dated or radiocarbon dated archaeological maize samples to understand that dispersal. The first example of directly dated maize for Peru is in the north at about 6,000 BP. The next most recent example is in northern Chile. So if you kind of zoom out here to the map to the right, that is about 2,000 years before present. Now, I should point out this is just published data. There are some studies going on right now about dispersal just north of this area as well. So as of right now, this map is showing about a 4,000-year gap of directly dated samples in Peru. Our study region of Moquegua in southern Peru lies just a couple hours north of the most recently directly dated example. So to understand how maize evolved and diversified in the Andes, we need to talk about the ecology and archaeology of the region. So let's zoom in a little bit. And on this map to the right, you can see Moquegua that's starred. And these little lines are rivers, so we're in a river valley. And we're also going to be talking about Tiwanaku, which is up here in Bolivia and on the shores of Lake Titicaca and at pretty high altitude. As we know, evolution happens within an ecological context. For the Andes, we need to start thinking in vertical. Around our study site is the coastal lowlands, which is the Atacama Desert and the driest desert in the world. Our region of Moquegua is a fertile river valley in the foothills of the Andes. So imagine a strip of green and a desertous mountain backdrop. If we go up the mountains, we enter the intermontane region where the highest yields of maize are grown today with terraced farming. And then finally, if we go up even further, we're in the Altiplano or the high altitude region. For us, this is Lake Titicaca Basin, where the lake is the highest freshwater lake in the world and lies at an altitude of 3,800 meters above sea level. So this is twice the height of the largest mountain in Colorado, to give you a little reference. The presiding archaeological theory is that little or no maize was being cultivated by early people at this high altitude. Maize textbooks today even indicate that the crop can't grow this high, but I visited this region this summer and I saw maize being grown, so we know it's adapted there today. So we know maize was important for those highland people, so where was it coming from? So the most interesting thing about our state region of Moquegua Valley is that there's 3,000 years of documented maize cultivation, and we know there's a relationship to the Tiwanaku. We think Moquegua Valley was likely a region controlled by the Tiwanaku because of maize-producing irrigable lands. So early cultivation in Moquegua Valley was through floodplain irrigation and subsistence farming by the Warakane people about 1000 BC to 600 AD. Now, you may think of this as kind of lazy farming, spreading out some seeds, letting the river naturally come up, flood the plains, and irrigate as they go. During this time in the Altiplano, something exciting was happening: the rise of the first statehood in South America by the Tiwanaku. Their capital city, also called Tiwanaku, was on the shores of Lake Titicaca, which is now Bolivia. The Tiwanaku people colonized Moquegua Valley in AD 600 and 1000 and used sophisticated irrigation canals to intensify maize cultivation. So it's likely maize was sent from the lowlands to the highland urban core to support the growing population, and specifically the Tiwanaku's thirst for maize beer, or chicha. The Tiwanaku collapsed in AD 1000 to 1300, and the Tumilaka moved into the valley. Several centuries later, we see the rise of the Inca Empire, and the Inca was also really interested in maize. They had cultivation centers where they imported soil types, and they used different irrigation techniques to develop desired germplasm, which they then dispersed throughout the entire domain to be grown. So what we think is happening is that when the Tiwanaku colonized Moquegua Valley, they colonized the Warakane, and then they were growing maize intensively and likely sending it up to the highlands. So what is the plan? What are we actually looking at? We hypothesize that preferred highland germplasm migrated with highland human populations to the lowland settlements, resulting in maize admixture and adaptation to this new context. Okay, so here's just a flowchart of the plan. We're going to be looking at the Warakane era. Remember, that subsistence farming. What we expect to see in the genomes from this area are an excess of lowland alleles, little highland alleles, and selection for traits based on that group's preferences. So what kind of varieties are we looking for? What do you think is going to happen? Well, we know from stable isotope analysis of human bone that there are small amounts of maize being consumed over the Warakane era. We also know there's ceramic evidence that maize beer, or chicha, was being brewed in pretty small amounts. The problem is, right now, with only measuring morphological features of maize fossils, it's really hard to tell what types of varieties were being grown and what was being selected for. So really, we're not sure what they were selecting for. And then over the Tiwanaku era, we expect to see an excess of highland alleles, decay of highland alleles over time, and selection for traits based on the group's preference. During this era, we know from stable isotope analysis of human bone, the Tiwanaku were consuming much larger amounts of maize. Through the ruins of large breweries in the city of Tiwanaku and large amounts of ceramic vessels, it's clear the Tiwanaku valued chicha. So it's likely the Tiwanaku had preferred varieties of maize that grew in the highlands, but it's unclear whether it was enough to fuel their needs. So we think they were colonizing the valley to grow specific varieties. And one of the varieties I think was being grown was for chicha. Today that looks like Murado or Kahuli, but really we don't know. So by comparing modern to ancient genomes, we may be able to understand what traits are being selected for. And one of the big picture things is, does that genetic legacy of the Tiwanaku era persist today in maize? I want to tell you a little bit about the materials that we plan to work with. So these are examples of maize macrofossils from a similar time, just a few valleys over. These cobs are different shapes, so square, round, and oval, with different cupule numbers and orientations. This may be a good indicator of the diversity to expect, but genotype to phenotype is really messy. So it's hard to say if this is going to be diverse or not. I do want to point out that while some of the cobs are charred and unusable for ancient DNA extraction, most seem to be in good whole condition. So we're hoping that ours will look like this as well. The materials we're going to use are housed at the Museo Contasoyo in Moquegua, and we have access to hundreds of samples with some from the Warikane era, so that early subsistence farming, and a lot from the Tiwanaku era and Tumilaka era. So we can be looking at the rise and the collapse of the Tiwanaku state. It's also worth noting that we feel pretty confident that the cobs contain decent ancient DNA due to the hyper-arid environment of the Atacama Desert. So overall, we should have plenty of samples to compare to modern maize in South Peru. I mentioned that this is a comparative genomics study. So what are we comparing these ancient samples to? Well, we're creating a modern diversity panel, and I want to tell you a little bit about how this came about. I spent the summer working at an archaeology site a few valleys over from my study site. This summer, we're waiting on the permit to be able to go into the local museum and look at some of these archaeological samples. So we decided to just walk around and talk to people and get the lay of the land. What does maize look like within Moquegua Valley? So I collaborated with an Indigenous grad student and soon-to-be licensed archaeologist, Elisa Pucar. Elisa is fluent in Quechua and Spanish, and really we just walked around and we talked to Indigenous farmers and elders. We decided to script what we talked about just for clarity, but honestly, they were pretty informal conversations. So we learned pretty quickly that Indigenous farmers and elders were excited to share their knowledge and wisdom with us. Most were surprised that we were interested in maize and not quinoa or potato, and they wanted to know why. So as they were sharing with us, I was sharing with them. I shared pictures of my home in Iowa with the landscape of maize and the varieties I grow in my own garden, expressing how maize is part of my own heritage and identity as an Iowan. So really, we were just having conversations, and often those conversations guided us where we should visit next or who we should talk to. So here's something we didn't expect. Over these conversations, we didn't expect to start accumulating maize. So we didn't ask for varieties, but they were often gifted. As we're talking to community members and explaining our study, they said, here, learn what you can, let us know. And so as we started getting varieties that way, we made a decision that if we're going to represent diversity of the valley, we should probably also be supplementing from the markets. So as we're learning about more varieties, we're trying to target those in markets and purchase those to make more of a broad diversity panel. I do want to point out, to prevent extractive practices, we did ask community members what questions our research can ask for them. While most really had no idea, we had one very testable question. We were asked, how does maize grow at such high altitude? So to help answer this question, we chose to do an altitudinal gradient and supplement our diversity panel with high altitude examples. I plan to share the results of our study with the communities. So I worked through the local agriculture office as part of our conversations, and I will be sending our results back to those stations for dissemination. In the lower right-hand corner, this is a snapshot of the diversity of maize I saw during the summer survey in Peru. So notice different shapes, sizes, and colors, and we're hoping this indicates high genomic diversity, but we won't know until we dig in. So at least phenotypically, this looks pretty exciting. And because I'm talking to a maize group, I would love to nerd out with you about the varieties I encountered this summer. One of the things that we learned was, at least in this area, maize is categorized by color first and its use second. So for example, white or blanco is usually used in mote, soups or stews, or choclo, boiled. Purple or red speckled, those varieties are called Cabanita, Chajeque, Chiquelo, Ceniza, Pisala, Misturita, and Plomo. And typically these are tostado or toasted and eaten as snacks. The yellow varieties are Amarillo, which is often used as animal feed. And one of the interesting things I learned about Amarillo is that it's not grown in Peru. It's usually imported from Argentina or the United States. So it must be very specific varieties that they're looking for. Chulpi was often tostado, but Chulpi was interesting because it looks like a sweet corn, but it wasn't talked about like a sweet corn or specifically used like we would use sweet corn. And then Pichia. Pichia is interesting because it was the only one that I encountered that was talked about as a sweet corn and grown specifically for one dish, pan de choclo, which is like a sweet corn cake. And all the places I visited talked about this variety as being just used for that one dish. So I thought that was pretty interesting. And then finally, solid purple. So this is Morado. And often this is used for chicha or fermented maize beer. So we plan to create a broad data set of diversity in the study region and in Peru to place the ancient samples in a pre-existing context. Currently, we have 70 samples. About 40 of those are from the summer survey and 30 are supplemented through the PI station. We were really intentional about choosing high and ultra high varieties to answer the questions asked of our research by community members. To understand coastal, intermontane, and altiplano traits, we chose a wide variety of altitudes over the north, the central, and the south regions. There was an enrichment in locations where the Tiwanaku state had a presence just to make sure we were trying to capture the diversity in that area. From the survey, I should point out we had many different named varieties, but there were quite a few that there wasn't a name for it. Often in the marketplace, whoever was selling those varieties were, this is a white variety or a yellow variety, but didn't specifically have a name for it. For those that we did have names for, when I was supplementing germplasm from PI station, I was trying to focus on new varieties as well, so different named varieties. One of the things I noticed in the survey was popcorn was never mentioned. I didn't really see it. We're not sure why, but there was a theme when I was talking to local people, and that was that diversity in maize varieties has dropped in the past 30 to 50 years. So it could be I just didn't see popcorn. Maybe it's not being grown as much. Maybe it's being consumed locally. We're not sure. But when I was trying to supplement our panel, I was sure to target some popcorn varieties as well. I do want to point out that I plan to use already available SNP data for comparisons. So for references, I'm looking at B73 just to be interesting, but really I plan to choose an Andean Peruvian variety to be a reference. I'd like to explore whether early samples are closer to Parviglumis, considering the short dispersal time to Peru. So I expect to see like drift away from Parviglumis allele frequency over time. And I'd also like to include an outgroup as I explore phylogenetic relationships between the varieties. So right now I'm looking at Tripsacum and Zea luxurians. And right now, I know this group is an expert in many of these genomes. So if there's things I should be thinking about here, please let me know. All right, so just to zoom out and show you a little bit about these locations. Our study site is located here in the south. There's a couple of maize stalks, realize it's kind of hard to see in the picture. And the Tiwanaku Highland City is represented here by a stone arch. So the people were migrating and trading back and forth between these two sites. So I tried to focus very much on that area during my summer canvas. All of the pins that are in red are examples from the summer survey, and the ones in white are from CIMMYT or PI station as supplements. I do want to point out that we focused on the north, central, and southern regions, and we focused very much on the southern regions because that's where the people are migrating in our study area. And we do have some interesting varieties. So we have some pretty ultra high altitude varieties. One I pulled from Amintani Island in Lake Titicaca, and it's likely we could see genetic isolation. I don't know. I mean, that's kind of what's exciting about this project. We're not quite sure what we're going to find. All right, so let's talk about how I'm going to pull this off. So what are my testable questions and hypotheses? Well, my first question is, what was the original source of maize in Moquegua Valley? what I think is happening is that maize dispersed along the coastal corridor shortly after arrival in northern Peru from Mexico and has an excess of ancestral lowland alleles. My second question, how similar is ancient maize to extant maize? I think early maize is more similar to the wild ancestor, but may align with modern maize, both in variety type and altitudinal gradient. And what traits were the Tiwanaku selecting for in Moquegua Valley? Okay, so I think subsistence maize in the valley was likely selected for lowland traits. When the Tiwanaku colonized that area, they introduced highland gene flow, and highland traits were selected against for adaptation to this new coastal environment. And then finally, the Tiwanaku were selecting for traits associated with chicha production because we know they valued that very much. Zooming out, here are the methods. There's a lot going on here, so we'll go through it slowly. All right, I'm going to start with the orange, the modern maize. We have 70 accessions right now with 20 locations. We are currently growing these out. As of this week, we've collected all of the tissue, and I'm currently isolating the DNA from that tissue. We plan to do whole genome sequencing at 2 to 5 depth coverage here at Iowa State University. And really, we're doing low coverage just because I'm focusing on alleles and we have a limited budget, so it makes sense for what we're doing. So our goal here with the modern maize is to create a diversity panel to represent the study region and Peru. All right, going over to the green, the ancient maize, we are hoping to export about 200 cobs. We're going to be doing our sequencing at Gregor Mendel Institute in Vienna. And we plan to do high-depth sequencing because we want this data to be usable for other studies, and the full process will ultimately be destructive. Right now, we're in step 2 of 3 to export those samples. And if you've been watching the news at all, Peru is experiencing some political turmoil, so we're being very patient as we go. The first thing we're going to do with those cobs is do a subsampling of about 30 in different contexts. What I mean by context are our soil stratum. And really those should be representing time periods, but we won't know until we do radiocarbon dating. So that first subsampling, what we're checking for is DNA quality. And we're focusing on about 5% endogenous DNA, which is typical for other studies like this. Once we have those contexts identified for where we expect to find high quality DNA, we're then going to choose about 100 samples, and we're going to directly radiocarbon date those at University of California, Irvine. We'll be doing mass spec for C13 and 14 for correction, and our goal here is to directly date all samples for the largest temporal timescale. Then we're going to sample the rest of the context, so about 100 cobs, and the goal here is to broadly sample the context identified for high quality DNA and the largest temporal timescale. And then at the end of this, our goals are to establish demography of ancient and modern samples, establish relationship between ancient and modern subpopulations, establish adaptation, gene flow, and signatures of selection over the Tiwanaku era. And I would like to zoom in and explore alleles associated with chicha varieties specifically. Okay, I'm gonna talk about the methods a little bit. This is where I'm feeling the least confident just because I am so early in my learning. So if you have feedback on this section specifically, please let me know. To tackle the first question, what was the original source of maize in Moquegua Valley? I need to establish demography of ancient and modern samples. I'm gonna be doing this through SMC++ and structure. What I expect to see from SMC++ is a drop in effective population size indicating the Andean bottleneck. I also expect a drop in effective population size with the introduction of Highland Tiwanaku varieties and low population size with increased genetic load. Over on the other side for structure, what I expect to see here when compared to the results from the Wong et al. 2021 study about high-low traits, I expect from the structure to see Warakani samples with an increase in genetic ancestry from a coastal lowland group. Oh, excuse me. All right, so the Warakani samples will have an increase in genetic ancestry from a coastal lowland group, and the Tiwanaku samples will have an increased genetic ancestry from an Altiplano highland group. For my second question, how similar is ancient maize to extant maize? I plan to establish the relationship between ancient and modern subpopulations using FST and SMART-PCA. What I expect to see from FST is high population differentiation from the Tiwanaku era samples compared to earlier Warakane era samples. This should indicate an aggression of highland material moving with the Tiwanaku people. I also expect to see a decrease in differentiation over the Tiwanaku era. Finally, I expect to see high population structure in extant samples, indicating adaptation to a range of ecological environments. What I expect to see from the SMART PCA is that Warakane samples might cluster near the Mexicana and Parviglumis, indicating early dispersal, but I also think they're going to be skewed towards northern extant varieties as well. The Tiwanaku samples might cluster closer to the extant highland samples, indicating that admixture. And the full diversity panel really should be spread out all over the place because we're talking about ecological and altitudinal gradients. And then finally, my third question, what traits were the Tiwanaku selecting for in Moquegua Valley? So I plan to establish adaptation, gene flow, and signatures of selection over the Tiwanaku era. I'll do this using admixture and PBE. So what I expect from admixture, and at the bottom of that graphic, I'm looking on the far right-hand side with the blue peak and the green peak. I expect to see the Warakane samples will have a similar proportion of domestication alleles to Mexicana and Parviglumis, indicating that early dispersal. The Tiwanaku samples have proportions similar to highland samples with selection for a diversity of varieties, and the extant samples will have most similar trait proportion as similar types of maize. And what I expect to see from PBE by comparing the high-low published data from the Wong 2021 study, I expect to see a de-enrichment of highland alleles in Warakane era samples. And after the Tiwanaku colonization, I expect to see a pulse of highland adapted alleles in Moquegua Valley. Over the Tiwanaku era, I expect to see a decay of highland adapted alleles over time as these would be selected against in the new ecological context. Furthermore, I'll be focusing in on differences in allele frequencies as the most extreme example should be important for highland and lowland adaptation. And then finally, I'd like to zoom in and explore alleles associated with chicha production. So while looking at changes in allele frequencies, I'd like to focus on alleles that could be associated with traits valued by the Tiwanaku people over the Tiwanaku era. We know the Tiwanaku produced high quantities of maize beer, and I wonder if we can identify candidate alleles by comparing contemporary Murado varieties to other varieties. So I want to identify other types of maize than Murado that are used in chicha today. So I have a handful of Murado from different regions right now, and I have one Kuhuli, but I'm also looking for other varieties that are used in chicha today. And I'm targeting about 8 to 10 different varieties with a range of locations. I'll be identifying candidate genes with functions related to chicha production. And specifically, I'm wondering, are similar highland alleles maintained in Moquegua Valley while others are lost? And could those traits that are being selected for be related to Tiwanaku preference for chicha varieties? And finally, broader impacts. So this is really a unique opportunity to explore ancient crop DNA at a region with 3,000 years of documented maize cultivation. I'll be able to elucidate evolutionary processes of the deeply diverged lineages of Peruvian maize. I'll be exploring how gene admixture reveals how maize moves with people, and what were those groups of people selecting for? And are legacies within maize of ancient human movement from the Tiwanaku state? And I will also be clarifying maize dispersal from Mexico to South Peru and shed light on the processes of empire building and colonization in South America. I would like to thank the research team. This is an interdisciplinary international collaboration. We have collaborators in Peru, Austria, and a great support team here in Ames. So thank you everyone for being part of this project. And then finally, references. I'll post these slides for you too in Slack if you want to look back and see what I was referring to. Okay, that was a lot. What questions do you have?
You want to start with Maud's question? Do we know which traits may have been selected for fermentation? What makes a better ear for fermentation? I guess you started to address that at the end, but
yeah, that's a great question. So I've only found one study on this that could be helpful, and this is coming out of a group from Brazil. I'll go back to that slide and show you which reference that is. Okay. Okay, so the group is Salvador Reyes 2020, and that paper that I'm referring to is Proving Andean Maize, General Characteristics, Nutritional Properties, Bioactive Compounds, and Culinary Uses. So they look into characteristics of different varieties of maize, and one of them was specifically Morado used for chicha. So they're looking at things like kernel moisture, sugary content, things like that. And I'm hoping that will give me a little hint on what to look for. But that's the only study I found that talks specifically about chicha. So if there's other works out there, please let me know about them. Okay.
Thank you.
You're welcome. All right, so I see Jeff's question, what evidence the Tiwanaku brought maize with them? That's a good question. We don't know. So here's what I'm thinking is happening. I don't know if you're familiar with, hold on, let me get the term right here. There's a book I'm reading now that's definitely influencing how I'm looking at this. So really, all of the works about the Tiwanaku talk about colonization. So we're talking about a change in power dynamic. The term I'm looking for is ecological imperialism. And this was coined by Alfred Crosby. And Crosby was talking about colonization. The dominant people bring cultivars with them and usually force them in a new ecological context where they're colonizing. So I'm taking this framework, and through that theory, I'm saying that I think the Tiwanaku are taking their preferred cultivars with them when they colonize Moquegua Valley. And they're colonizing that region because it's a fertile river valley, so they're able to grow a lot more maize than they can in the highlands. So that's the theory I'm going with. But actually tracing the varieties, we don't really know because, you know, the macrobotanical evidence, you have kernel or cob characteristics measurements, but that's it. We really don't know.
And Heather, if I'm remembering right from our conversations with Andrew, there's pretty good documentation of a shift in the way agriculture was done in the region when the Tiwanaku colonized. So
yeah, absolutely.
Yeah, the assumption is that that shift was accompanied by new varieties.
Yes. Thanks, Matt. All right, next question from Denise. Did you find the land race called Chocoseno? It is thought to be... No, I didn't find that one, Denise. Do you know if it's more of a southern variety? One of the things I noticed was the different names for varieties depended on where you were. Like the name for one thing in the south would be an Aymari version of that name versus the central or north would be a Quechua version of that name. Do you know?
Oh, Heather, just check the chat below because we have someone here who knows exactly where Chocoseno is from, and it's Colombia with very high rainfall. So yeah, I unlikely that you would find it in your Peruvian sample, but I always find things that are called Tripsicoid, I find very interesting, of course, and I have grown it, and it is pretty crazy.
How does it grow? What does it
look
like?
Well, of course, you grow it in Ithaca, it's meters and meters tall, but it is considered to be sort of like a primitive maize that is cultivated directly into slash and burn. You know, it's like the first thing they plant into a newly created field. So I was just curious if you had it included in your diversity panel.
Interesting. Thank you for sharing that.
Yeah.
All right. I see Raphael talks about color as another trait. Absolutely. So I'm hoping from how maize is classified today by color first, kernel color first, we're hoping we see the same sort of thing historically. Again, we don't really know, but that's one of the things we're looking into. Miguel's question, how does the frequency of ancient maize remains change in the different archaeological strata? Yeah, so the frequency, the Warakane era, the subsistence era, there's a lot less than there is over the Tiwanaku and post-Tiwanaku eras. Okay, Fausto, 200 ancient maize samples, all from Moquegua Valley, or include other areas? That's a great question. So the 200 ancient samples, they're specifically in Moquegua Valley, and they're in slightly different locations because each of those different locations are different things. They're almost always middens, which are trash piles. So we see, you know, many different cobs within the same area. Some of those are slightly in hillsides. There's also one place that is, we think, ritually important. So there used to be a Warakane temple in that area and a Warakane burial site. And then when the Tiwanaku colonized that area, they built a temple over the Warakane temple. So I'm really interested to see if there's more ritualistic maize in that area or something specifically selected for that's different than others. I'm not quite sure, but each of the sites are slightly different.
Maybe something to keep in the back of too, Heather, that there's the Sama Valley samples, if we ever want a comparison to Moquegua. So there are archaeological specimens that are out there from nearby valleys as well.
Mm-hmm. And I should say that Sama Valley is 3 valleys over, and the Tiwanaku presence was less in that valley than in ours. What's really interesting about Moquegua is that we have that 3,000 years of documented maize cultivation and archaeology from the Warakane, the Tiwanaku, the Tumilaka. It's really interesting site.
Heather, can I make some comments?
Please, yeah.
Yes, so the title of your talk is like the first state in South America. So this is something that Tiwanaku is a very advanced civilization. I don't know, but from one of my understanding, there is an ancient city in the coast of Peru, Caral, that is I think it's the oldest like civilization that has been like dig out of Peru, but I may be wrong, but because Tiwanaku are pretty advanced. They have canals, they have all this technology out there by the Titicaca area. So I don't know. Well, just I was surprised. Another thing is, in your first slide, you show us like the Moquegua Valley by the Yungas on the west, and it's a little bit confusing. So maybe you can change the Moquegua Valley that it goes into the Pacific coast instead on the Amazon basin. Another thing about the choclo, that word choclo, at least in Colombia, we would use that word specifically for sweeter corn, sweeter yellow corn. I don't know if in Peru, choclo designates some classification of different corns, because we in Colombia, we use it usually that corn is like raw and sweeter than the corn you use for making the local tortillas that are called arepas. So that word may be used different, and it's from a Quechua word. So that's a different word. I don't know the usage, but that might be interesting because you were talking about the usage in chicha, that it should be sweeter because it will be good. It's easier to ferment, probably. And that's it. I have a lot of things. Maybe I would like to write to keep in contact. Yeah. This is a super interesting project. It's very nice samples. Super nice.
Thank you. Feel free to send me a message in Slack. I can add my email. My email address might be in our schedule too.
Yes, another thing about Caral is because this colonization probably was the coastline. The corn should easily go through the coastline rather than the mountains, and so that's another interesting thing. Yeah, so a lot of things. Thank you.
Yeah, Pastor, we should talk. I think we have a lot of good things we can talk about.
Pastor, are you going to be at the maize meeting?
I cannot be there.
Oh no! I was thinking you and Heather should have a long conversation. Sounds like to talk about.
Okay, it looks like Maud has a couple more questions. Oh, the reference for the colonization. Yes. Okay. So this was Alfred Crosby, and the book is Ecological Imperialism. And then Maud also had a question. Were the cobs used for fermentation or the stalks? Do we know? That's a great question. We don't know for sure. So we're using our framework of how chicha is brewed today and looking backwards. And typically the cobs are left out to start fermenting. So the kernels stay on the cobs. So they start sprouting a little bit. So as I'm walking around in the markets, you see cobs that look like they're starting to rot, but really they're starting to sprout a little bit. That's one of the first steps in fermentation. But I only saw the cobs being used as that. And as I ask people how they're making chicha, it's always with the kernels on the cobs first, like that's the first step. But we're not sure if the stalks are used. So that's a great point. We're not sure.
Okay, thanks. Yeah, I know that in some other crops, sometimes they use the stalks or the leaves or whatever. So, okay, but if they use cobs, at least for sure, it's an important organ, right, to study. Okay. Mm-hmm.
All right, are there any other questions? Having sweet stalks, yeah, I heard that as well, especially with the really early varieties, if they were selecting for the stalks versus cobs or kernels, yeah. And if anyone has information about fermentation or chicha production, like Fausto, you mentioned the Blanco varieties. You know, I did see white chicha this summer. That would make sense. I'm trying to learn more about that too, to know what sort of traits I could be seeing in the genome.
Yeah, I mean, Heather, as you get sequence, I mean, we know in sorghum what makes sweet sorghum, and a lot of that stacks up. So you know, you should be able to look in the genome to see if there's mutations that are likely to produce sweet stalk corn. So
yeah, that's a great point. I'll add that to my notes here too. Be thinking about the sweet sorghum.
Ed, has anybody scanned just the GBS data or something else to look to see if any of those are present?
I don't. Yeah, I think you want to just kind of. Does anybody know what the mutations are for sweet sorghum? Yeah, I can't remember what's if it's the starch to branching or what it is that builds up the sugar in there. So so maize is not sugar cane, but if you wanted to make it sugar cane, you could. Probably without too many mutations.
All right, any other questions or comments?
Okay, seeing none, I think I want to invite everybody for next week. Jeff Rasabara will be speaking. Same time, same place, same links. But thank you very much again, Heather, for presenting today.
Yes, thank you everyone for being here. I appreciate it.
Thanks, Heather.